Morning, everybody. My name is Kaylee Byers. I am the chair of the Wildlife Diseases Working Group of the Wildlife Society. I'm also an assistant professor in the Faculty of Health Sciences at Simon Fraser University in Vancouver, Canada. And I'm calling in today from the unceded ancestral and territorial lands of the Musqueam, Squamish, and the Tsleil-Waututh nations and peoples. And to get us started, I just wanted to thank all of you for coming. And this is really exciting. This is the first TWS webinar that I've introduced as chair of the Wildlife Diseases Working Group. Our group is large. We have over 300 members and seems to be growing every day. And we've had a lot of exciting work going on recently, looking at upcoming workshops and seminars for TWS. So if you're not already a member, have a look at the work we're doing, but it's a lot of fun. And I will pass it over to Chair-elect Kim to introduce today's speaker.
Thanks everyone for joining. Today we're welcoming Dan Greer from USGS to talk about a student network for amphibian pathogen surveillance, acronym SNAPS, which is building a sustainable disease surveillance network across North America. So SNAPS leverages student power to conduct ongoing Bsal surveillance by incorporating surveillance into undergraduate curriculum. To help overcome challenges with underfunding for early detection surveillance. So the surveillance program addresses the challenges of traditional approaches while providing a unique educational experience for students. And Dan Greer is a research biologist at USGS National Wildlife Health Center in Madison, Wisconsin, where he works on a variety of wildlife disease systems. Among those systems, he spends significant time addressing disease threats to amphibians as part of the USGS Amphibian Research and Monitoring Initiative as a co-lead for the North American Bsal Task Force Surveillance and Monitoring Working Group. And after Dan's presentation, Oliver Hyman and Xavier Jackson are going to share perspectives from an instructor and student view. And this presentation is pre-recorded, so Dan and his colleagues will take questions at the end. So I'll turn it over to Dan. Thank you, Dan.
And if you do have questions, you can put them in the Q&A at the bottom of your screen. There's a little Q&A button. You can type them in there. Do we have you okay there, Dan? Oh no, Zoom is freezing. Of course, classic.
Okay, I think it's back. And I'm going to share screen.
Wonderful, we see slides.
Hello, good morning. I'm Dan Greer. I'm a biologist at the US. Program. This webinar covers a lot of facets: emerging diseases, conservation and management of non-game species, surveillance, teaching, and probably some more that I forgot for now. And I'll be going through the background behind all this, but the underlying motivation is a problem-solving exercise where a group of researchers, educators, and managers together to solve a conservation problem.
Technology doesn't want to be our friend. Looks like Dan is reconnecting to audio.
Oh no. Okay. Did it kick me out?
Yeah, it's being fairly glitchy.
Okay.
Yeah, so far we can't, at least on my end, I can't hear the presentation very clearly.
As someone who hosts
All
a
right.
lot of events, this is really classic.
Yeah.
I'm gonna give it one more try.
Okay.
And then we will see about me just going through the slides without the audio live.
Okay. Thanks, Dan.
And like I said, I'll be going through the whys and the whats and how we got here. But our program has a pretty straightforward mission. It's to mobilize students to lead the search for emerging amphibian pathogens. And what we want to see in 5, 10 years, or even longer, is a sustainable experiential learning network that drives early detection of emerging wildlife diseases. First, I want to give a short background that highlights why the focus on amphibian pathogens. Over the past 20 years or so, diseases had an outsized impact on amphibian biodiversity, with just one and now two chytrid fungal pathogens having global impacts that includes widespread population declines and even significant contributions to species extinctions around the world. And as a wildlife science and conservation community, we have and continue to try to deal with emerging pathogens in a data-deficient world. The emergence of pathogens into global disease is the result of rare events, and in the case of amphibians especially, the invasion and spread of the chytrid fungal pathogens has been rapid and really caught us unprepared, and just stitching together retrospectively what happened and what the consequences have been. With our current focus on Bsal, though, we are starting out a little more prepared, because a common pattern in chytrids, as well as several other amphibian pathogens, is that the global trade of captive wildlife is moving these new diseases around the world along with their hosts. That's a fact of the world today that creates a dynamic threat, but it also gives us some information to start from. Okay, so time for our fungal biology lesson for the day to give more background into the diseases and pathogens we're focused on. So the amphibian chytrid fungus pathogens have a unique life cycle that depends on an amphibian host, but also has complex ecological and environmental requirements. The primary concern for our frog here is that BD or Bsal make their living on the skin. In amphibians, the skin is an absolutely crucial organ that is responsible for many crucial physiological functions. But the fungus also depends on aquatic or at least very moist environments. The part of the fungus that infects the host grows and generates infectious life stages called zoospores that are released from the skin and causing disease in the process. Zoospores are free-living and can reinfect the same host or transmit to a new host that they encounter in water. So there are two known chytrid fungal species that are pathogenic to amphibians, and both cause skin infections. Batrachochytrium dendrodebatis, or BD, is fairly well known. It was one of the earliest wildlife diseases that was documented causing severe impacts to frogs and toads by causing mass mortality, impacting populations, and likely has been a cause of species extinctions globally. At this point, BD is endemic in most places in North America. And we, the amphibian species, biologists and managers, are living with the threats and the impacts that BD cause, and have a reasonable understanding of the negative impacts that BD can cause and how they're variable across different amphibian species. Bsal, Batrachochytrium salamandrivorans, is the new cousin to BD. It's really truly an emerging disease that's a global threat. Bsal has only been discovered in the last 10 years or so, and it's probably not present in North America, but there's much uncertainty surrounding its extent and severity of the impacts if it were to be introduced or established in North America. Bsal was discovered when biologists in Europe started to observe severe declines of fire salamander populations that they couldn't explain. That led to the characterization of an entirely new pathogen and fungal species that was given the name salamandrivorans, or devourer of salamanders, because of the high mortality and necrotic skin infections it caused. With further investigation, the emergence of Bsal in Europe was linked to the global pet trade. Bsal was identified on several brightly colored, and really cool amphibians that are popular in captive collections and as pets from Asia, notably a group of species that are collectively referred to as fire-belly newts or fire-belly toads. And so this detection occurred in native wild populations in Southeast Asia, where Bsal is present but does not appear to cause disease, and also in captive collections, where researchers were able to piece together strong links among the occurrence of Bsal and exchanging animals among these captive collections across the European continent. And that spread through captive amphibians is one half of what makes Bsal such a threat to North America. There is and has been continued imports of Bsal susceptible species into North America. There have been some positive regulatory and captive industry reactions to the Bsal threat, which would cover another seminar or three, but the introduction threat remains. And to put some context on that, before Bsal was discovered, there was ranged anywhere from in the tens of thousands of captive salamander species like these fire-bellied newts imported into the US every year. Similar magnitude for fire-bellied toads. And to put some additional perspective on there, other very common captive amphibians like African clawed frogs in the genus Xenopus, or common frogs in the genus Rana are imported in the tens or even millions of live animals per year. So the other half of the threat to North America is that we are a global hotspot for salamander biodiversity, and many of our species are highly susceptible to Bsal through experimental trials. North America, and particularly the Southern Appalachian Mountains and Pacific coastal regions, have an incredible diversity of salamanders, with about 30% of the world's species occurring in North America. We have three completely unique groups of endemic salamanders: mole salamanders, amphiumas, and sirens, and an incredible radiation of Plethodontidae, or lungless salamanders, with about 150 unique species occurring in North America. Add to that several of our most common and widespread salamander species, western newts in the genus Ptericha, and the eastern newt or red-spotted newt are highly susceptible to Bsal disease. This means that if Bsal establishes in North America, there are potential consequences both for rare and unique species, but also a likely loss of common and very ecologically important species in our newts. The North American response to Bsal was to establish a task force with federal and state agencies, university researchers, conservation NGOs, zoos, and the pet trade to create a network of working groups to try to prevent and be prepared for Bsal in North America if it were to be introduced. That led to a series of initial actions, including risk assessments, regulatory actions on salamander imports, and an initial large-scale surveillance effort, because initially we didn't even have any idea or any way to answer, was Bsal already here in North America? After all, the organism was just discovered a few years earlier. There are also a variety of interesting research projects and communication efforts going on. And if anybody's interested in getting more in-depth information on what has gone on and what is going on, the Bsal Task Force website is at salamanderfungus.org and has lots of great information. But since this webinar is focused on the surveillance part of things, we did not detect any Bsal in North America in an initial really big surveillance effort. Over 2 years, we collected over 11,000 samples, led by the U.S. Geological Survey's Amphibian Research and Monitoring Initiative, focusing on high-risk areas and species. And so it's great that we didn't detect Bsal. But that brings us around to the problem I presented up front. This was an expensive, time-consuming surveillance effort, and there's not funding or capacity to coordinate another one. It's hard to maintain surveillance for early detection of something that isn't here yet, especially when we have already stretched resources for wildlife disease surveillance from diseases that are here, for example, avian influenza or white nose syndrome. And the amphibian conservation and management world also has existing threats to deal with that almost certainly have to take priority, for example, habitat loss and invasive species. And now we're going to turn and focus on the solution to the problem that we're trying to establish, SNAPS, the Student Network for Amphibian Pathogen Surveillance. It's really a great fit to try to establish and maintain surveillance for an emerging disease for a couple reasons. Particularly, amphibians are easy and safe to capture, and Bsal and BD are both cutaneous or skin pathogens, so the sampling is easy. All you have to do is take skin swabs. And amphibians and diseases present a range of learning opportunities for students. It can be easily incorporated into coursework with conservation, environmental policy, microbial ecology, disease ecology, herpetology, and epidemiology. So the goals for SNAPS are to establish a wide-ranging, ongoing, coordinated, and sustainable Bsal surveillance program, to coordinate this dissemination of Bsal surveillance results so that people know where we've looked and hopefully where we haven't found it, and to provide a diversity of students with an engaging course-based research experience. And it really boils down to pairing surveillance objectives and learning objectives, and putting those two together to leverage one with the other. The SNAPS is run through the Surveillance and Monitoring Working Group within the North American Bsal Task Force, where a subset of members forms a steering committee that helps make decisions and guide the growth of SNAPS. This also makes sure that the activities of SNAPS are coordinated with the other North American Bsal Task Force working groups. SNAPS is funded through the U.S. Geological Survey in Environment and Climate Change Canada, with quite a bit of support from the Amphibian group. We currently run diagnostics through two labs in the U.S., the National Wildlife Health Center and the University of Georgia at the Southeastern Cooperative Wildlife Disease Study. And in Canada, it's coordinated through the University of Guelph. We've also have a recent successful spin-off where a more focused SNAPS program is being developed in the state of Minnesota, funded through the University of Minnesota's Aquatic Invasive Species Research Center. So where we are is we're a network and we're growing. We currently have 54 participating institutions, 42 in the United States, 10 in Canada, and 2 in Mexico. We've developed a logo website and post results and lesson plans on our webpage, snaps.amphibiandiseases.org. And want to emphasize that the network is really trying to be North American, not just US based. So our colleagues in Mexico are helping to translate materials into Spanish, same with colleagues in French speaking areas of Canada, so that it is a truly North American effort. Our operational goal for SNAPS is to make it as easy as possible for instructors and students to participate, from the onboarding all the way through obtaining and assessing results, both of the surveillance and learning. With sign up, we provide sampling guidelines, biosecurity protocols, assistance with animal care and use permitting and site access permitting. Sampling materials and shipping is all free of charge. The instructors then have a student learning pre-assessment that we ask them to administer to their students, and then they teach. We have several lesson plans established to go along with SNAPS, but instructors are also free to incorporate it however they see fit into their courses. And of course, we're always interested in having examples and sharing examples of how it's been, how the SNAPS experience has been used to teach. Then comes the fun part, which is catching, swabbing animals and sending samples. So this is the real experiential part for the students. And after the students collect the samples, they send the samples to the diagnostic labs where we use standard molecular tests, quantitative PCRs that both detect Bsal and BD DNA. And we aim for a quick turnaround on these tests. That's one of the points of emphasis so that instructors can return the results to the students so they can both see what they did and potentially use them in class. All of our results are open. We have a project page on the amphibian disease portal hosted by Amphibian Web, where both other researchers as well as other classes participating can see what the whole program achieved. And so that was, a really fast, high-level summary of how it works. But now I'm going to hand it over to one of our steering group members, as well as instructor who's incorporated SNAPS into his teaching for several years, Oliver Hyman.
Hello, my name is Oliver Hyman, and I'm a lecturer in the biology department at James Madison University. I'm here to speak about the instructor's experience participating in the Student Network for Amphibian Pathogen Surveillance, aka SNAPS. Before I get into the details of implementing SNAPS, I want to first provide a little bit of context about James Madison University, where I teach. JMU is a primarily undergraduate institution with a little over 20,000 undergrads. As you can see on this map, JMU is located in Harrisonburg, Virginia, about 2 hours southwest of Washington, D.C., near Shenandoah National Park and the Appalachian Mountains. A great place for sampling salamanders, conveniently enough. Our biology department is one of the largest at JMU with over 4,000 majors. Currently, freshmen in the honors section of our introductory biology course, like the ones you see in the image here, participate in SNAPS as part of the several authentic research experiences that are embedded in the lab portion of their intro course. This is the timeline for my implementation of SNAPS at JMU. I reached out and was accepted to participate in SNAPS in the summer prior to my first implementation. The following fall, I began formally planning for implementation for the coming spring semester. This entailed participation in a brief orientation webinar, perusing freely available SNAPS lesson plans and protocols available through the SNAPS website and a shared Google Drive folder, scouting local field sites to ensure I could find enough salamanders to sample, obtaining local collection permits, and completing IACUC training at my university for amphibian sampling so I could be added to the SNAPS umbrella IACUC protocol. With all that in place, I ran SNAPS the following spring with my freshman students. This entailed getting us sampling and shipping supplies free of charge from SNAPS, tailoring teaching materials for my needs, and reviewing the sampling protocols much more closely so I could gather all the necessary materials and work out the logistics for getting my 24 students out into the field to sample salamanders for BD and Bsal and home in time for their next class. This is a more detailed timeline of exactly how I implement SNAPS in my 24 student, 2 hour and 50-minute freshman biology lab course. On the very first day of class, I make it clear which day we will be going into the field and that everyone needs to mark that day in their calendar. I typically have them reserve one other day just in case the weather doesn't cooperate. I'll also recruit 1 or 2 students to drive a departmental vehicle on the day we go into the field. I then break up the SNAPS experience across 3 lab periods. The first is a 2-hour session where I provide a background lecture and we discuss one seminal paper about Bsal. This provides students with a decent foundation to understand why we are sampling salamanders. After that, we do a quick primer on working in the field that covers basic supplies, attire, and expectations, and then watch a short but detailed video that covers the SNAPS sampling protocols, which SNAPS provides. Lastly, I remind students exactly where and when to meet next week so we can promptly hop into our departmental vehicles and head out to the field. Time is tight the following week, so we meet in the parking lot, load right into the vans, and hit the road ASAP. I typically show up a bit early to load up snacks and supplies. I'll drive one vehicle and get student volunteers to drive the other vehicle or vehicles. We are lucky enough to have a field site that is only 20 minutes away with incredibly high densities of eastern spotted newts. So we can drive to the site, set up shop, sample the 20-30 animals in about an hour to an hour and a half, decontaminate our equipment, eat a little snack, take a few photos, and we'll be back to campus with time to spare before students need to get to their next class. All the protocols, vials, swabs, shipping supplies are provided conveniently by SNAPS for free. All I need to do is just have some very basic field supplies like markers, scissors, gloves, buckets, and bleach. Depending on when we return to campus, I ship samples out the same or next day and typically get results back in about a month, at which time students and I will spend about 50 minutes in class sorting and analyzing the data. SNAPS has been an incredibly positive experience for my students. It's taught them about the negative effects infectious pathogens can have on host populations, and it's given them solid experience in the field, handling live amphibians, and contributing to a real-world conservation issue. These are potentially resume-worthy experiences that may help students develop a career in conservation or wildlife biology. SNAPS is also beneficial to wildlife managers and the salamander species they protect by providing low-cost, semi-regular, continent-wide Bsal surveillance. The black dots in this map represent SNAPS partner institutions that have classes and instructors just like me who are helping to sample for Bsal in many of its hotspots for potential introduction to North America, as depicted in dark purple. Lastly, SNAPS has also been great for me as an instructor. With its ready-to-go lesson plans, easy-to-follow protocols, and free supplies, it's been a cheap and easy way for me to incorporate a great lab experience into my course. It's also enabled me to kill two birds by combining my teaching and research, which bolsters my annual reviews with cool course-based research experience and provides fodder for grants and publications like the one you see here, recently released by Olson, along with several SNAPS instructors in the journal Frontiers in Amphibian and Reptile Science. Perhaps most importantly, everyone, including myself and all the students that I've taken out into the field and other instructors I've talked to who have done SNAPS, has really had a blast getting their hands dirty and going out and interacting with real animals in the field. I feel very fortunate to be a participant in SNAPS and want to sincerely thank the people, sponsors, and students who make it all possible. Thank you.
And now I want to go over some of the results we've had so far from our early pilot efforts in 2020 through having about 50 schools participate in 2023. This includes a summary of some of the numbers and the sampling where we've been doing and what we've been doing, an example of the student experience, and then some results from an initial assessment of the learning aspects of the SNAPS program. Through 2023, we've had 52 different academic institutions participate, including in Canada and Mexico, and probably with the new instructors and courses participating this spring, we're probably, I think, close to 70 different institutions participating. That's resulted in over 1,200 students participating, collecting over 2,500 samples from nearly 50 different amphibian species, and we haven't detected Bsal. And like I mentioned before, our results are all posted to the amphibian disease web portal with a link on the screen, or you can use that QR code to navigate there if you're interested. And these are a great set of overall numbers, but what was really valuable about what we're getting out of this is what the students are able to learn and get from this. So now I'm going to have Xavier Jackson, a student who participated at Tennessee Tech University, share some of his perspectives.
Good morning, everyone. My name is Xavier Jackson, and my pronouns are he, him, his. I'm a student TA and research assistant in the biology department at Tennessee Tech University, and I'm here to kind of speak about my experience participating as a student in the Student Network for Amphibian Pathogen Surveillance, also known as SNAPS. Before I get into the details of my experience with SNAPS, I want to kind of provide some context about Tennessee Tech University. So TTU is a primary undergraduate institution with around 9,000 undergrad students. Our university is nestled in between multiple state parks, as well as being only 2 hours away from the Great Smoky Mountains, which is a biodiverse hotspot that boasts 2 endemic salamander species. The SNAPS process for students is relatively simple. First, they need to attend a pre-sampling lecture to learn about chytrid fungus, as well as how to prevent spread. From there, the group is usually divided to collect specimens and sample them accordingly. After that, we send the samples to be tested using qPCR and await test results. Our team took 2 trips, one in the fall of 2021 and one in the spring of 2023. For these trips, we chose City Lake Natural Area in Cookeville, Tennessee. This site in particular has a large population of plethodontid salamanders, which are important due to their subcutaneous respiration, making them at higher risk of contact pathogens. As you can see in this figure, all samples from fall 2021 tested negative for BD, Bsal, and ranavirus. However, when our team sampled again in the spring of 2023, we unfortunately had a single adult dusky salamander, scientific name Desmognathus fuscus, test positive for Bactochytrium dendrobatitis, or BD. This is the first ever positive test result at this site, and this is going to indicate that continued surveillance is crucial to mitigating the spread of chytrid fungus and other pathogens. And there are so many ways in which students can benefit from the SNAPS program. Not only does this help students obtain real-world field experience, it increases scientific and wildlife disease literacy, exposes students to tangible STEM pathways, and gives students multiple opportunities to make direct impacts to their environment. Personally, I feel as though participating in SNAPS has given me the confidence I needed to be able to see myself in the field of scientific research. Not only did this program help actualize my passion for herpetology, it also helped me discover my desire to protect and conserve sensitive ecoregions. Without SNAPS, I would not have had the opportunity to help me build the confidence I need to succeed. Shortly after receiving our results, I was invited by Dr. Aubrey Hill to present the logistics of our pathogen surveillance at the first annual Global Amphibian Reptile Disease Conference. Being able to work closely with researchers around the globe for this event helped open my eyes to new perspectives early on that I would not have been able to receive had I not attended this conference. I was privileged enough to win second place in undergraduate poster presentations, and I couldn't be more grateful for the opportunity. I've also had the privilege of attending the Tennessee Herp Society conference as a guest and participate in a population survey for multiple amphibian species. Aside from that, the applied knowledge that I've learned from this position have helped me gain other positions in the field. For example, I worked last summer as a research technician in the Tennessee Tech Aquatic Invertebrate Lab. I've been able to assist in future sampling surveys with incoming graduate students and be able to teach others about the dangers of chytrid fungus and other zoonotic pathogens. Thank you all for giving me the time to speak today. I want to give a special thanks to SNAPS, GUARD, and most importantly, Dr. Aubrey Hill, who really took a chance on me. And if it wasn't for her, I would not be here speaking to you all this morning. So thank you all so much for listening, and I hope you have a great day.
The last aspects of SNAPS I want to present is an initial assessment of student learning outcomes. We can measure the surveillance side of the program through how many swabs you have and how many detections and numbers like that. But it's just as important to be able to evaluate the student learning goals, both so that we can demonstrate success and also so we can see where we can improve to make the student experience the most successful. This learning assessment was led by a master's student, Jennifer Perez, and her advisor and SNAP's steering committee member, Jenny Walk, as part of Jennifer's master's thesis at Eastern Washington University. The learning assessment included 4 categories of student outcomes. What were the impact of SNAPS on students' effective outcomes, like with questions about their enjoyment of nature, their self-efficacy towards environmental conservation, and career interest in STEM. What was the impact of SNAPS on students' cognitive outcomes, including content knowledge and knowledge of amphibian sampling methods in the field, as well as their confidence as measured by self-reported content knowledge. What were the SNAPS outcomes consistent across multiple institutions? The answer to that is yes, and I'm not going to go into any results there. And last, how equitable were the outcomes of SNAPS, which is very important to assess how well SNAPS is reaching and engaging different groups, student groups. The assessment was conducted by asking undergraduate students to complete an optional pre-survey and post-survey after participation in SNAPS. This included 294 survey responses in 2022. All of these were anonymous and de-identified. And that resulted in 70 paired student pre and post surveys across 9 institutions used for the results I'm going to share. Those included Auburn University at Montgomery, Eastern Washington University, James Madison University, Monroe Community College in New York, North Carolina State, Queen's University, the University of Guelph, Warren Wilson College in Western North Carolina, and Washington State University, Vancouver. This is the makeup of the student respondents for the learning assessment. About 2/3 were female. About little more than 70% included traditionally represented student ethnicities in higher education. The traditional demographic groups that were considered underrepresented in STEM higher education were included Armenian, Asian with Hispanic, Latino, and Spanish origin, Black or African American, American Indian or Alaska Native, Hispanic, Latino, or Spanish origin, and white with Hispanic, Latino, or Spanish origin. About 2/3 of the respondents and participants included upperclassmen, including 3rd or 4th year college students. And it was broken down about by half whether or not the students' parents had attended college or not. When it came to the impact of SNAPS on the students' effective outcomes, They generally had high scores in terms of career interest in STEM, enjoyment of nature, and self-efficacy towards environmental conservation efforts, although there was a detectable effect after participating in SNAPs and reporting a higher self-efficacy towards environmental conservation efforts. The students' cognitive outcomes were quite a bit more varied. The assessments showed that we could have detectable effects and that the students learned about amphibians, Bsal, and how to sample through SNAPs. And also both in terms of content knowledge questions and self-reporting, these cognitive outcomes increased. When it came to how equitable SNAP's outcomes were, underclassmen, so first and second year college students, had greater gains in the content knowledge after participating in SNAPs compared to upperclassmen. When it came to assessing how equitable the outcomes of SNAPs were, we did detect that first generation college students had greater gains in content knowledge compared to the non-first generation students. And it also appeared there were some probably pretty complex potential patterns in gender and ethnicity, although it'll take additional years of surveys to better tease those kind of relationships out. To wrap up, we've made some good progress towards our goals of integrating wildlife pathogen surveillance and experiential student learning, but we also have plenty of challenges ahead. The first take home that I don't want to get lost in the details is that we are doing something proactive for a wildlife disease threat, and that's really pretty unique. As wildlife disease researchers, managers, biologists, we usually find ourselves reacting to something after something bad has already happened. So this is a unique situation to try to make some positive impact into what could be a pretty severe disease threat. Plus, this is a pretty fun activity, and it's really easy to connect and see how it's empowering the next generation of scientists and biologists. One of the challenges is always finding resources to keep up with growth. SNAPs, the concept of SNAPs shifts some of the cost and coordination for surveillance away from traditional and agency-funded projects. But it's still not a magic solution for the dollars and hours that go into building and maintaining, particularly a continental scale wildlife disease surveillance network. So proposal writing and chasing funding is always going to be crucial because we want SNAPs to grow. And we have had some promising growth, particularly with one example being the spinoff in the Minnesota SNAPs project by integrating with invasive species interests. Last is the challenge of increasing diversity and reaching more underrepresented student populations. Our initial learning assessment showed how learning outcomes from SNAPs can be impactful, and one of our goals with growth is to include more student groups that are traditionally underrepresented in STEM and higher education. And that involves both being able to reach those students in their institutions and also finding ways to reduce barriers to entry for the instructors who might participate in SNAPs. And so one of the ways we're trying to do that is to increase the numbers and types of learning modules we have available so they can fit into many different course types. So I hope you enjoyed, maybe learned something from our experience trying to build this student-powered surveillance network. If anybody is interested in becoming involved, there's a couple different ways. Course instructors, if you are interested and want to do something right away, that QR code will take you to a Google Forms link where you could enter some contact information and background. And although we do not have any more capacity for SNAPs classes in 2024, if you're interested and fill out that form, we'll contact you right away when we look to start planning out for 2025. If there are any students out there who think this is great, please share this with your professors. and faculty and see if they're trying to get them interested in participating. If they're agency managers or biologists, a great way to get some surveillance and to connect with, again, the future generation would be to find potential university and college partners in your area and direct them to SNAPs. We'd love to see more agency integration with the universities who are out there. And then anybody who's viewing this and is interested, we're always looking for more participants in the North American Bsal Task Force. You can email me at the email up there if you have interest in joining the Surveillance and Monitoring Working Group, or you can check out the other working groups and other ways to get involved through the North American Bsal Task Force at salamanderfungus.org. And with that, I'll wrap with questions and discussion. This QR code takes us to our SNAPS web page with more information. And then the slide is some of my favorite parts of participating in SNAPS, particularly as a federal agency biologist who doesn't get to interact with students much. At least I get to see all these different classes and students out there who got to participate. So thank you.
Thank you for that, Dan. Wonderful. We managed to get through the technical hurdles, so that's fantastic. We do have about 12 minutes here for some questions, and maybe I'll ask the first one, and then Kim, I'll throw it over to you. But we have a few questions from Samantha, and so maybe we'll start here with the first one, which is, what's the difference between North American and Mesoamerica, and is there any account for the amphibian importance in Central America and Asia?
So I think, yeah, Mesoamerica is generally referring to more subtropical and tropical areas on the continent. And at least in our program, we're, certainly not trying to neglect other parts of the Americas or the world in terms of amphibian biodiversity. But our focus has been to build and try to get ahead of the Bsal threat to North America. And certainly amphibian researchers are aware, and I don't really know exactly what type of activities are going on in other parts of America, other parts of the world where Bsal may be a threat, but I'm confident that they're aware of it.
Okay, thank you.
You want me to ask the next question in the chat?
Yeah, go for it.
So how is BD endemic? I thought it was introduced from Asia. That's the second question we have.
Yeah, that's another seminar or 5. Certainly the highly pathogenic genotypes of BD had genetic introduction from Asian strains. But as far as considering BD endemic in North America, what we're finding, and particularly through some of the sampling in SNAPs, is that it is in a lot of places and in a lot of species where it's not apparently causing any kind of disease or health issues for the amphibians individually or at the population level. So at the very least, it sure seems to be widespread and coexisting with its host populations. Certainly it's still causing some problems in particularly some Western frog species, but on the whole, it's been here long enough, probably been here longer than we think, enough to consider it, having endemic pathogen population cycles.
So I'm scrolling down a few questions, Kim, because we have some questions at the bottom here about the program. So I thought maybe we could shift to those. So you mentioned like beyond 4th year. So what does that look like? Is that like people who are doing like I did in my undergrad, like a victory lap, like year 5, or is there like into graduate school? What's that look like?
Hang on, I found the question. Yeah, that's graduate. So some of the instructors are doing this with graduate students as part of, as part of graduate classes. So that's what young fourth year is. So potentially some super seniors maybe. We kind of left it up to the instructors and the students to classify where they were in terms of their college journey and stage.
Thanks.
And Brandy wonders if there's any interest in universities who want to do the qPCR themselves to save some financial costs. Is that an option in the program?
It is something we are exploring as far as growing goes. There's a couple of, I don't want to say challenges, but considerations we're taking in terms of this, particularly with having the potential to find a high consequence conservation disease out there. For the time being, our preference is to try to centralize into labs that participate with the Bsal task force, and one of the other working groups, the diagnostic working group, maintains some quality control testing on a periodic basis. So for the time being, we're limiting the labs involved to labs that participate in that network with the Bsal task force, but it is something we're discussing with different schools and different instructors who teach and have particularly courses based on molecular techniques. So we're considering it, thinking through the problems, and benefits, because it would be a great benefit to distribute that kind of thing.
And we have one more question here about the program, and if anyone else has any questions, please feel free to stick them in the Q&A. We do have a couple minutes left. Do you know if institutions also try to incorporate iNaturalist into SNAP's efforts?
I do not know if anybody does or not, but I agree. Yeah, it would be a great way to pull in different bits of information for different research efforts or for different types of teaching about populations or abundance, or try to measure hard to find things.
Great, thank you.
And another question just came in. Does the SNAPS program absolutely need to be part of the undergrad course, or can the student chapter of the Wildlife Society organize this program in partnership with a professor?
No and yes. I think it'd be great to have, for instance, student chapters reach out and organize something like this. We do want to recognize that part of the formal classroom training doesn't necessarily come with that type of thing, but it hits just about everything we want to do. So if whoever asked that question is part of a student chapter who's really motivated, reach out. We'll talk about it. We do have a handful of environmental education centers participate as well.
Great. Well,
Great.
I think that's all for questions.
I've got some questions if I can ask. I really enjoyed your presentation, Dan, and I appreciated that you looked into some impacts of the program on students. And I was curious if there have been students that have been stimulated to go on to graduate school from your program, or have done if there's opportunity to do undergraduate thesis projects, using some of the data from the network, if that has been a thing you've noticed.
Not that I've noticed. We're always trying to compile that type of information. I can think of one student who participated at with collaborator at St. Cloud State University in Minnesota, who's now entered a master's program interested in the disease aspect of amphibian conservation. And then as far as incorporating data into student theses or research projects, absolutely. That's part of the goal is, and our open data effort is that it's all out there, right? It's there for people to use.
Great. Yeah. And I was also curious if you, I'm sure you've done plenty of analysis on the structure of the surveillance system, but in your view, do you think it's sufficient to detect the introduction of, a disease that would be rare at first? And what do you think could be some current gaps? What are you trying to strengthen at this point to improve that early detection capability?
So no, it's probably not. If we're thinking about this Bsal threat at the continental scale, the SNAPS, however many students we can get out there is is probably never going to be enough to detect an initial very rare introduction of something. But it's more than would be going on otherwise, which is nothing. And so for part of what we do is try to focus recruitment for a couple things. First is for engaging particularly underrepresented students in STEM. So finding institutions that aren't your usual R1 research type schools to do this. But the second is trying to steer and recruit schools that occur in the high risk areas that include a couple different groups, best assessments of where it could be introduced, and probably more importantly, where the species that would be most impacted are. But at the end of the day, we just want people out there to get something. And it might be a long-term accumulated thing to start building confidence about not detecting or being able to detect it if it did get introduced. So it's small steps trying to accumulate towards, something on the surveillance side of things that's really
Yeah, I think that's a
valuable.
good vision.
Yeah.
I was wondering too if you think this system, like I think your salamander system has some attributes that make it more readily implementable in this framework of student involvement. Do you think there are some other systems where something like this in wildlife health could work well?
Yeah. I mean, I don't see why something like, monitoring small mammals wouldn't fit well. Other small things that are easy for, relatively easy with little training for students to do safely for the animals and themselves. Another avenue that we've discussed and we're thinking about in growth, but really don't have time to think about it hard, is to incorporate environmental DNA sampling into a network like this. It comes with its own challenges, but also some interesting benefits, both for, again, teaching and being able to monitor a lot of stuff passively.
Yeah, potentially a good one for HPAI.
Mm-hmm.
Thanks. Well, those are all the questions I have.
I think we're close to time, but there's one more question that I think we'll ask because I love a policy question. So this is from Sarah. If these pathogens or other health threats are identified through SNAPs, is there a policy advocacy or management suggestion aspect to the program?
Not through SNAPs specifically, but yes, through the integration with the Bsal, North American Bsal Task Force. So that group is writing and always revising response plans and communication plans. So in the event of a detection, the right people can know quickly and start doing something about it. And then slightly more loosely related to the surveillance part, but the Bsal Task Force also has a clean trade and captive animal working group trying to work with commercial sellers and traders of captive amphibians.
Great. Thank you so much. And thank you everyone for coming. Thank you, Dan, for taking the time, and to your team for presenting. I don't know what else to say other than I hope everyone has a lovely rest of their day. Really.
And weekend.
Good weekend. Once I hit close here, it's going to end our session. Thank you again, everyone. And Dan, again, thanks for coming out for the Wildlife Disease Working Group.
Absolutely. Thank you.
Webinar series.
Thank you.
Everyone.